All right, we're going to move right on to our next speaker. Looks like it's ready to go here. So Dr. Michal Minczuk is speaking from Pretzel Therapeutics. The title of his talk is Mitochondrial Genome Engineering in Vivo.
Hello, can everybody see my screen?
Looks good.
Yes.
Yeah, thanks very much. So thanks very much for this kind introduction, and also to invite me to give an opportunity to talk about our recent data. And although I'm involved in Pretzel Therapeutics, I'll mention that today I'm having a University of Cambridge hat on. Here are my disclosures. So before I get into the main subject of the talk, I'd like to give you a little bit of background why the mitochondrial genome is, in particular in mammalian systems, is very difficult to modify, to engineer. So at the moment, we don't have any efficient ways of delivering DNA into the organelle. So this genome is quite small. In principle, could be synthesized entirely, and then we could deliver either modified version or deliver normal version in order to correct mutations mitochondrial DNA. But at the moment, this is not possible. CRISPR-Cas9, which is obviously a very useful to modify the nuclear genome in mammalian systems, hasn't been adopted to operate in mitochondria yet. So as a consequence, we have very few models that would precisely mimic mitochondrial DNA-associated diseases. This is contrary to the nuclear mutations that Vamsi already mentioned. However, there are some positive. There are established methods to change existing heteroplasmy, meaning the mixture of mutant and wild-type molecules within a cell. And there are emerging tools to precisely edit particular bases in the mitochondrial genome. And through the rest of my talk, I'll be concentrating on these 3 points. So the question we've been asking for over a decade now in the lab was, how can we take a sick cell which has pathogenic heteroplasmy, meaning the mixture of wild type, so green molecules here and mutant red molecules here, and can we shift them into a more favorable ratio, meaning that we'll have more wild type molecules compared to the mutant. Why would you do that? Because if we have enough wild-type molecules, we'll be able to move the cell beyond the pathological threshold and basically restore its function. And we call this process heteroplasmy shift. So how do we do it? Us and others in the field came up with an idea of creating specific cuts in the mutant mitochondrial DNA. And then because the cells have pre-programmed copy number, so the cell sense how many copies of mitochondrial DNA should have in it. These, after cutting the mutant, the cell will repopulate itself, mainly wild-type molecules. And the nice thing about this procedure is that you don't have to eliminate all the mutants. You have to just move it beyond the pathological threshold. Over the years, we developed several ways of cutting mitochondrial genome precisely. These tools are called programmable nucleases. So nuclease is an enzyme that cuts DNA because you can program it in order to bind any DNA sequence, hence the term. So there are zinc finger-based nucleases where you can manipulate these colorful boxes here in order to design new DNA-specific enzymes. Other nucleases involve tail DNA binders, so called mitotalens. Also, most recently, there was a paper showing that also very old tool called meganuclease could be also adopted in order to manipulate the mitochondrial genome. So how does it look in practice? Our lab studied mainly zinc finger nucleases. So now I'm presenting an example of a mutation, 8993T2G, which is often associated with neuropathy, ataxia, retinitis pigmentosa, and also if the mutant level is higher, maternal inherited syndrome. So in order to bind this mutation, we designed a pair of zinc fingers. The red one binds specifically to the mutation. The green one here is a supporting one, and then when these two the so-called monomers come together, this is where the DNA is cut precisely in this point. In normal molecules, because the red monomer doesn't bind to the sequence which is not mutated, this cut doesn't occur. So we experimented with this system for in cells in culture. We have taken a heteroplasmic cell line, which with about 80% of mutant. This cell line have a and has a reduced oxygen consumption rate and also low OXPHOS complexes. And upon treatment and optimization of the zinc fingers, we managed to shift heteroplasmy from about 20% wild type to about 80% wild type. When we analyze these cells by proteomics analysis, so looking into steady state levels of the protein and oxidative phosphorylation system, we noticed that this cell was basically rescued. So yellow means more OXPHOS, blue is where this oxidative phosphorylation system is slower. What we also managed to do was to recover oxygen consumption, and altogether we managed to rescue this this cell line. So the next important step was to try this technology in vivo. Now, as I said at the beginning, there are very few mouse models of mitochondrial disease that in particular would be heteroplasmic, so would have a mixture of wild type and mutant. However, our collaborators in Cologne developed a mouse model with a mutation in one of the tRNA genes in the mitochondrial genome. So this is sort of part of the supporting system of the oxidative phosphorylation. And in high level of heteroplasmy, this mouse model developed a rather mild but cardiomyopathy. So we decided to experiment on this animal because we didn't have any other ways of proving that our system would work in vivo. And then we designed specific zinc fingers that would bind and cut the position which was mutated in the mouse 5024. Then encapsulated DNA coding for this zinc finger nuclease into a AAV vector. This was a AAV9 subtype, AAV945, which has tendency to go into a heart, so cardiotropic, and is heavily detargeted from the liver. This is where AAV viruses like going. And this is a little bit busy on the right-hand side, but what we observed that depending on the dose of AAV vector use, we managed to shift heteroplasmy in this animal. It was safe in terms of not reducing a copy number of mitochondrial genome. Also, longer treatment led to a better result. Because the mutation in this tRNA gene renders this tRNA unstable, we looked into whether stability of this molecule would be improved upon treatment, and this is indeed what we observed as well. We also, using metabolomics, which was already described, we managed to rescue a particular biomarkers of mitochondrial dysfunction. I'm showing here lactate to pyruvate ratio, which is also quite often used in the clinics. And this technology was spun out to a startup company called Pretzel Therapeutics. Pretzel launched officially last week. Please have a look on the website to read more about it if you are interested. So for the remainder of the talk, I'd like to concentrate on the following questions. So can we develop mouse models with mtDNA mutation, which would be relevant to diseases, and then various therapeutics could be tried preclinically, including obviously maternal inherent Leigh syndrome, relevant for this conference. So I'd like to say that at the moment, mitochondrial genome engineering is undergoing a real revolution. About 2 years ago, there was a report of using cytidine-based editing, meaning that we could take pretty much any C in the mitochondrial genome and change it into a C. And on the opposite DNA strand, this will result into G2A. This was a collaboration between University of Washington and also the Broad Institute, and VAMSI also was collaborated on this very important paper. Soon after, other types of base editors were invented. So the first ones were based on tail as DNA binding portion, but also zinc finger-based cytidine deaminases, cytidine editors were introduced as well. And this year, another type of base editors was reported, allowing for A to G mutations. However, at the moment, this tool remains a little bit less precise as compared to the cytosine editors. So basically what we want to do, we would like to take a healthy cell and then edit a particular base in DNA so that this cell will become a dysfunctional due to accumulation of mutated mitochondrial genome. So quite the opposite what we've been trying to do for many, many years. Obviously, if we are very precise and efficient, this could also yield a 100% mutant, so-called homoplasmic cell lines too. These tools are also exciting because then the heteroplasmic mutations could be repaired by a cutting. However, the homoplasmic mutations, where all mtDNA molecules are mutated, were not, couldn't be repaired by cutting, because there is no normal DNA. Therefore, these base editors are quite exciting for future potential precise correction of homoplasmic mutations as well. So as a part of our efforts to knock out every single gene in mouse mitochondrial genome, I'd like to present one particular mutation which we introduced. I have chosen this one because it's relevant for the Leigh syndrome. I think mutations ATP6 gene in the mitochondrial genome is the most frequent cause of the Leigh syndrome, especially in the one which is maternally inherited. So our approach here was to select a codon early on in the ATP6 gene, and it would be a tryptophan codon. And then rather than DNA coding for this particular amino acid, we converted into a stop signal, a stop codon. So from TGA to TAA by editing cytosine on the opposite strand. So after lots of tests in vitro in cells to make sure that our constructs are precise and don't edit any other portions of the genome, we selected our favorite construct and then introduced it into a mouse embryo by microinjection, and then we implanted this embryo into pseudo-pregnant female. Our founder animal was at more or less 10% of mutation heteroplasmy. But then we could start selective breeding. So basically we could mate this female with the wild type normal male and look into the offspring and select females with the highest level of heteroplasmy. Obviously the mutation has to go through the germline. But we were lucky enough that this mutation went through the germline, and then we subjected this strain through selective breeding. And then, as you can appreciate from this graph here, in each population, in each generation, we were able to have a higher and higher level of heteroplasmy. So at the moment, we are at generation 4, and then we started to characterize this mouse. And then I'm showing here animals with these levels of heteroplasmy between 65 to 72. This is the analysis in skeletal muscle. And what we can see that the complex in which ATP6 is located, so-called ATP synthase, it's not intact. So it shows these quite typical subcomplexes which are typical for dysfunction. And these sort of subcomplexes were also found in patients affected by mutations ATP6, especially the mutations that truncate the protein. So as the next steps, and I have to admit, I'm very grateful for Pure Mito Foundation, and also Pure ATP Foundation. We'd like to follow up the studies of this animal, and then in collaboration with Stephen Gray and Alexandro Prigione, who will be our next speaker, we'd like to characterize this mouse. We'd like to apply heteroplasmy shifting technology to this mouse as well. We'd like to collaborate with Steve so that AAV therapy could be tried, and also collaborating with Alessandro so we can apply the base editing technology in induced pluripotent stem cells and organoids. And with that, I'd like to thank to all my team, especially Pedro Pinero, who's been leading the base editing program, whereas the zinc finger shifting was mainly run by Payam Gamic, but also I'd like to thank other collaborators and all the institutions for paying the bill. Thanks very much.